Welcome to Chapter 22, Microbial Diseases of the Nervous System. This chapter fulfills Objective 4. I'm sure you're very familiar with it by now. In this chapter, we're going to discuss structure and function of the nervous system, bacterial diseases, viral diseases, fungal diseases, and protozoan diseases of the nervous system, and we're going to finish off with prions. Now on to the structure and function of the nervous system. The nervous system consists of two separate parts. The central nervous system consists of the brain and the spinal cord and their associated membranes, and the peripheral nervous system, which consists of all of the nerves that come off of the spinal cord. Now, part of what we're going to be talking about in this chapter are meninges. These are the membranes that cover the brain and the spinal cord, and when they become inflamed, that is called meningitis. Now that we've talked about the structure, on to the function of the nervous system. The nervous system functions in bringing in information to the central nervous system and taking information from the central nervous system out to the rest of the body. So, say for example, I see a lovely red rose. That's information that's coming into my eyes, is being transferred from the nerves to my brain, which processes that information. The brain sends impulses to my legs to walk me over, sends impulses to the nerves in my back to bend over, and I sniff the rose. You get the idea. Under the topic of bacterial diseases of the nervous system, we're going to talk about meningitis, tetanus, and botulism. Meningitis, as I mentioned before, is inflammation of the meninges. Symptoms of meningitis generally start out as not terribly scary. You have a headache. You can have a headache from all sorts of things. Stiff neck. You can get a stiff neck from sleeping on it wrong, from an injury, and also fever. But when you see those three, you have to be careful, because meningitis, especially bacterial meningitis, can move really fast. Now, before we move on and talk about bacterial causes of meningitis, which are the most severe forms, I do want to mention viral meningitis just real quick, even though we're not in the viral part. Viral meningitis is caused most of the time by viruses in the enterovirus family. They're gastrointestinal viruses that can get into the blood and occasionally can cause meningitis. Generally speaking, viral meningitis is more mild than bacterial meningitis, and we just give supportive care and let the patients, recover on their own. Bacterial meningitis, on the other hand, can progress rapidly and is more often going to be fatal than viral meningitis, and that's the one, the cause that we're going to be most concerned about. There are 4 types of bacteria that can cause bacterial meningitis, and we have vaccines for 3, so their incidence has lowered. The first one is Haemophilus influenza. It's an opportunistic pathogen. It's a normal inhabitant of the upper respiratory system, but it can get in and cause meningitis. And another one is Streptococcus pneumoniae, and it's also an opportunistic pathogen that we have a vaccine for. The third one that we have a vaccine for is Neisseria meningitidis. Now, because we've taken care of these 3 main causes of bacterial meningitis, there's been a 4th one we've seen greater prevalence, and that is caused by Listeria monocytogenes. Listeria, generally speaking, causes mild disease in adults. If you have a suppressed immune system, or if you're a fetus, women who get Listeria monocytogenes while they're pregnant, oftentimes there can be problems with the fetus. Either there can be a miscarriage or a baby born with birth defects. Listeria is a foodborne illness that can get into the blood and cause meningitis, which is why I mention it here. There hasn't been a movement toward creating a vaccine for it. Instead, we've been working on decreasing the levels of Listeria in food. Now, Listeria has a tendency to show up in prepared foods, especially cold cuts. They've been popping up in cold cuts quite a bit lately, and also in dairy products. But they're working on ways of getting rid of all the listeria or reducing the numbers that somebody with a normal immune system can handle. Now, with all four of these bacterial types of meningitis, if you catch them quickly, when you have those first three symptoms, headache, stiff neck, and fever, we can treat them with antibiotics. If you go on to high fever, and this can progress to convulsions and coma, usually by then with bacterial meningitis it's too late and the patient dies. Now, before we leave meningitis, there are other causes of meningitis, especially in those who are immunosuppressed, but we're going to leave that discussion for another time. Tetanus used to be a terror. Also known as lockjaw. Tetanus is caused by Clostridium tetani. It's a gram-positive obligate anaerobe that if you get the endospores into the deep tissue, they get in, replicate, produce toxins that cause clenching of the muscles. Like, I think this is the most famous picture of a tetanus victim. This is a British soldier, and I believe about the 1700s, and you can see his whole body is just in one major Charlie horse, which just sounds absolutely horrible. In fact, there have been spasms of the back muscles that have been so bad that it snapped the spine of the patient. Tetanus is a situation where replication of the organism is not a problem. It's producing the toxin that's the problem that causes the disease. So nowadays we have a vaccine for it, where you take the tetanus toxin, inactivate it with formalin, forming what kind of vaccine? That's right, a toxoid vaccine, and it gives us protection. Now, if you ever step on a nail or have a deep puncture, get your tetanus shot updated. There's no sense in risking this kind of thing. Botulism is a cousin, if you want to call it that, of tetanus. It's caused by Clostridium botulinum. It's in the same genus, and it's also a gram-positive obligate anaerobe that produces endospores. And it also produces a toxin. Replication of the bacteria within your system isn't what causes the disease, it's the toxin. In this case, the toxin also attacks the nerves, but it does the exact opposite. Instead of causing the muscles to clench and not release, it causes the muscles to not receive nerve signals. So you go limp. In fact, you go so limp, you stop breathing and you die. Now, botulism is considered to be a foodborne disease because that's generally how you get it. You bring in your produce from the field, it's covered in dust, covered in botulism spores, and if it's canned incorrectly, you create perfect anaerobe environments. You've got food, you've reduced the other bacterial competition, and the endospores come out, and you get all sorts of botulism replicating in this airless environment, producing all sorts of toxins. Well, the toxin is scentless, tasteless. You don't know you've got it until you're passed out on the floor. And unfortunately, in this picture, this was most of a family that was killed by eating home-canned green beans, and just sad, sad situations. So make sure if you're doing home canning that you do it properly. And if you see a bulged lid, don't even mess with it. Just toss it out. It's not worth it. It can also show up in infants. You've probably heard that you shouldn't give infants honey. Well, this is because botulism endospores are everywhere, including in honey. And we generally pasteurize honey to get rid of most of the bacteria, but pasteurization does not get rid of botulism endospores. Now, when an adult eats honey and is exposed to the endospores, our immune system has seen botulism endospores before and takes care of them before they start replicating a whole lot. If you're talking a child under the age of 2, sometimes those endospores can germinate and replicate within the baby, producing toxins, and it causes floppy baby syndrome. Basically, it's botulism from having a botulism infection. Botulism endospores has also been linked to sudden infant death syndrome, SIDS. So don't give your kids honey. until after the age of 2. By then, their immune system has matured enough that they can handle it without a problem. Now, we can't leave botulism without talking about Botox. You're going, if botulism is such a horrible toxin, and it is, why are we injecting it into our wrinkles? It's all a matter of dosage. If you get it in large amounts, in uncontrolled amounts, by the organism replicating, it'll kill you. If you get very, very small dosages, controlled, that's being injected, then it causes your muscles to relax and the wrinkles to go away, and it's not a problem. Lest you think it's just for our vanity that we use Botox. Botox has been a method that has treated spasms of the vocal cord. There's a disease where the vocal cords spasm uncontrollably and it can get so bad that the person can't speak. Injections into the vocal cords causes those spasms to relax and the person is able to speak. And so folks with this disease go in for regular injections of Botox. Anytime you have muscles that are tightening up and not relaxing like they should, we can inject a little bit of botulism toxin and take care of the problem. Now on to the viruses. We're going to talk about poliomyelitis, rabies, and West Nile virus. Poliomyelitis is one of the most paradoxical of diseases. We didn't see it until there was an outbreak, at least in the United States. We didn't see it until there was an outbreak in the summer of 1894 in Vermont. You're going, huh? That late? Well, what they think happened is, before the advent of indoor sanitation, most people were exposed to poliovirus when they were still protected by their mother's antibodies. And so it was an asymptomatic disease. Later, when we developed indoor plumbing, most people weren't exposed to it until they were in their later childhood, early teens, when they no longer had their protection of the maternal antibodies, and it became a much more severe disease. The normal progression before the advent of indoor sanitation is you would pick it up from drinking infected water that had been fecally contaminated. It would get into your intestinal mucosa, replicate, pass out through the feces to the next person, and you either didn't experience any symptoms, or if you did, the symptoms would be headaches, sore throat, fever, nausea. Sounds like the stomach flu, doesn't it? But when you get it later in life, it can attack the nerves and cause paralysis. And in this picture here, we have folks who are in iron lungs that are providing ventilation for them so that they can breathe because the muscles of the diaphragm have been paralyzed. If you didn't die from not being able to breathe, or then it could attack the nerves of the limbs and leave you crippled. In fact, that's mostly what we think of as polio, is the paralysis that is caused when it attacks the nerves. One thing to keep in mind, though, even at its height, before we had a vaccine for polio, most people got the milder form. It was only about 1% of people who contracted poliovirus that became paralyzed by it. But you know, when you've got tens of thousands of people getting poliovirus, then that 1% is pretty high. So in about the mid-40s, 50s, we were able to develop a vaccine for polio. The first one was a killed vaccine, which is inactivated. But after that, we were able to come up with a live vaccine, and that decreased the incidence of polio to the point where we hardly ever hear of it anymore. In fact, there was a push, still is actually, a push to eliminate polio from the world like we did with smallpox. But we haven't been able to do that so far, mostly because, being a non-enveloped virus, it can survive in water for long periods. Basically, your best method of fighting poliomyelitis is vaccination and wastewater treatment. Now, you've all heard of rabies. It's been a long time since we've had outbreaks of mad dogs, mostly because we have, you guessed it, a vaccine. We vaccinate our dogs so they don't transfer it to us. Now, when the main reservoir of a disease is animals, then we call it a zoonotic infection, and rabies is a zoonotic infection. Let's talk about what happens in the patient, be it a dog or a bat or a human, and then we'll talk about treatment. So the virus is injected into the person's body, and it makes its way to the nerves and slowly makes its way up to the brain. Now, when it starts replicating in the brain of the patient, they have certain symptoms. They start having troubles moving, they stagger about, they have a hard time swallowing. That's what leads to the frothing at the mouth of the mad dog. Because they're having a hard time swallowing, they develop a fear of water, hydrophobia. In fact, that's another name for rabies, because, well, it's also a neurological side effect of it. Aggression follows as the neurons in the brain are damaged, and this leads to, in wild animals, lack of fear of people, alteration in their behaviors, and they're more likely to bite, which transmits the disease. Now, because we have a good vaccine for dogs and cats, we don't see it in our pets as much anymore. Mainly we see it in wild animals. If you see a carnivore or a bat moving toward you and acting aggressively, get away from it. If you are bit by this animal, try to keep a hold of them so that, and this is kind of blunt, so we can cut their head off and do immunofluorescent staining of the brain so that we can see if they're infected with rabies, and we can give you the vaccine. Now, you're probably thinking, isn't it a bit late? If I'm bit and I've been infected, it's too late for the vaccine. Well, actually, because rabies is such a slow-moving virus, we can actually vaccinate you after the fact. Once you start showing symptoms, though, the vaccine is not terribly effective because it's reached the brain and it's replicated to the point where it can start doing damage. It's better to get it as soon as you're bit. We've improved the vaccine. It used to be that it was a horrible regime of multiple injections into the stomach. You had a lot of side effects, fever, all sorts of nasty stuff. They're much better now. They're not very good. It's still not a pleasant vaccine, but it's a whole lot better than getting rabies and dying. Before we go on, let's mention the bats. We think of dogs, wolves, foxes, their weasel relatives like raccoons having it, but actually the main carriers of rabies, at least in this area, are bats. So if you see a bat during the daytime, avoid it. It's probably got rabies and is just waiting to bite you. West Nile virus, as you are all aware of, was introduced to the United States not too many years ago. It's now become endemic in all of the continental United States, and it's a relatively mild disease. Most people, it's asymptomatic, and those that do get symptoms, most of us are going to experience flu-like symptoms, fever, headache, nausea sometimes, but there are those that can develop encephalitis from it. That's inflammation of the brain. As you can tell, inflammation of the brain is never good. It causes nerve damage that can persist long after the body has taken care of the virus and you're virus free. So that's why we're concerned about it. They're working on a vaccine for it for horses. But let's talk about the life cycle of West Nile virus. West Nile virus is carried by, yep, you got it, mosquitoes. The mosquitoes bite us, okay, and inject it into us. It replicates, and it's a dead end there. In if the virus had its way, the mosquito would bite a bird. Here we've got our magpie here, member of the crow family, and it would replicate in the bird. Another mosquito would come, bite the bird, it would replicate in the mosquito goes and bites a non-infected bird, and it continues in a normal cycle. When the mosquito bites humans or horses, it's a dead end. We do not pass it from human to human by mosquitoes. In fact, when it was first moving across the country from New York, we could track the march of the disease by the death of the birds. The birds here are not used to it, and it kills them. Over time, the birds that survive West Nile will be more resistant, their offspring will be more resistant, and we will see change or evolution in process. Well, to wrap up West Nile virus in humans, it's a relatively mild disease unless you're very young, you're very old, or you're immunosuppressed. Now I'm only going to talk about one fungal disease, Cryptococcidius. This is, generally speaking, a disease that we only see in the immunocompromised, but it has become an indicator disease of those patients that have active AIDS or are moving into active AIDS who are HIV positive. It causes meningitis. It's a fungal meningitis. Now, one of the ways of diagnosing it is it's got a huge capsule. Look at that. Here's the fungus in there, and there's this huge capsule. So if you have a patient that comes in with meningitis symptoms and you pull a sample from their spinal fluid and you see these huge capsules, you know not to treat them with antibiotics. You go straight to the antifungals. And like most meningitis, other than the viral, it can progress very quickly. So you want to go with a proper treatment as quickly as possible. Now let's talk about protozoan diseases. African trypanosomitis. It's caused by the trypanosoma. And isn't that a beautiful pathogen? Isn't it cute? You don't want to get it, but you have to admit it's cute. It's carried by another biological vector, the tsetse fly, and we only see it in sub-Saharan Africa. And because that's where the tsetse fly lives, and it's called river sickness because that's where the flies like to hang out, where it's nice and moist. They come and bite people or cattle, and these lovely trypanosomes get into your system and they move into your nervous system, and you progress to coma. That's why they call it sleeping sickness. Once you move into coma, traditionally there is a very, very high mortality rate. Now, part of the problem that we've had with treating trypanosome diseases is they undergo antigenic variation. So what happens is you're infected with the first clone, okay, and you have a peak, your immune system attacks, drops down. Along about this time you have a switch in antigens, and you get this up and down, and progressively becomes worse. And so you get it the first time, you're sick, you get better, it comes back, you're more sick, you get better, comes back, you go into a coma for a short time, you get better, and it just gets progressively worse and worse until you die. This also makes it very hard to develop a vaccine for it. In fact, so far as I know, we have not been successful in doing this. Mainly, the way to treat it is vector control, get rid of the tsetse fly, and we also have some good antiprotozoal drugs for this. The problem is, though, most of them contain heavy metals, arsenic and what have you, so they have some severe side effects, but it's nothing like the damage that is caused to the nervous system by infection by the parasite. Now on to diseases that are caused by infectious proteins, which for a long time we had a hard time figuring out what was going on. The most famous prion disease right now is bovine spongiform encephalopathy, which, to translate the Latin, which is cows with spongy encephalo disease. In other words, their brains start looking like SpongeBob SquarePants. And let's talk about what's happening. In fact, there are the holes in the brain that you see at the histological specimen. What happens is there are proteins in the brain that function in the normal functioning of the neurons. But if they become the prion shape, they no longer function how they're supposed to. In fact, what they do is they become enzymatically active in going around and changing the functioning proteins into proteins that are now prions. And then they spread and change the functioning proteins into the non-functioning proteins, prions. And this goes on to the point where the neuron is no longer able to live and it dies. And that's where you get the holes in the brain. As this spreads from neuron to neuron, as these proteins march from neuron to neuron. Now, as the brain is damaged, as it starts getting holes, as it dies, the symptoms that you start seeing, you start having problems with movement. In humans, you start having problems with cognition, memory problems, problems with speaking. Makes sense. As your brain's falling apart inside of you, you start generating almost Alzheimer's-like symptoms and eventually progressing to death. Let's talk about where we think the prions came from in mad cow disease. There is a slow progressing neurological disease in sheep called scrapie. And the symptomology in sheep for this, as the brain becomes affected, they develop repetitive behaviors such as scraping up against posts or trees, trunks or branches, and they would scrape off their wool, and that's why we call it scrapie. Now, it was isolated in Britain in the sheep population, but what we think happened is it jumped to the cow population by the practice of feeding cows waste meal, meat products that humans don't generally like to eat. Brains, neural tissue, doesn't have a high commercial value, but if you feed it to cows, it's easier to digest than grass, and they put on weight faster. And we think that's how the prion disease got into the cattle population. From there, we saw about late 80s, mid 90s, we saw a high increase of a normal human, if you want to call it normal, a human neurological disease that we were aware of called Crutchfield-Jacobs. And this Crutchfield Jacob variant disease progressed much faster, whereas Crutchfield-Jacob generally went in families, progressed over years. You generally didn't start seeing symptoms until you were in late middle age. In the variant forms, it was hitting people in their 20s, and they were able to trace it back to this bovine spongiform encephalopathy, which has become known as mad cow disease. So we think that it jumped from the cow population into the human population, and since in Europe and in the United States we're not in the habit of eating people, it hasn't spread from human to human. But it has caused us to keep an eye on our food supply, and we don't process downer cows. These are cows that we find lying out in the field. They're unable to get up. We do not put them into the food supply anymore. In Britain, they have stopped the practice of feeding sheep meal or cattle meal to other cattle, and they think they've got it under control, but there's lots of people that have these prions that are slowly eating holes in their brains, and the symptoms have not shown up yet. Let's go back to the human disease, Crutchfield-Jacob. Okay, where did the prion come from in the human line? For that matter, where did it come in the sheep line? What we think happens is that there is a mutation in the DNA that causes the alternate form of the protein. And this alternate form of the protein slowly spreads from neuron to neuron, causing this progressive neurological disease. When it went from cattle to humans in Crutchfield variant, we think what's happening, the reason why it's progressing so much faster, is because you're getting a higher dosage. You're eating more prions in one go instead of making your own a little bit at a time. So we're keeping an eye on our cattle population. We're testing them for this on a regular basis. It's only been isolated in Washington in 2 different herds. We never made it into the human food supply, but we're keeping an eye on it. Also, part of the thing that worries us about it is it's really hard to kill these things. You can't kill them in the body. There's no treatment for it once you become infected. And also, from surfaces in meat products, prions are heat resistant. You can't cook them out. They are resistant to radiation. Even if you sterilize your foods with gamma radiation, that doesn't take care of prions. They are resistant to acid. When you eat them, the acid in your stomach does not denature the prion, and for whatever reason your intestines take these prions up and they eventually end up in your neurons in your brain. The only way to get rid of them is to completely incinerate the prions so that you're breaking those amino acids apart. They're working on enzymatic treatments so that enzymes can go and snip apart those amino acids. So far, those are the only ways to treat prions, that and don't eat them in the first place. Well, that's it for this chapter. Once again, I've got summary charts that were really good, but keep in mind, unless I talked about them in the PowerPoint, or if they're on the worksheet, you don't have to worry about them. Go over this as many times as you want, and come to class with questions.